Hello friends, we are discussing enzymes and proteins that are involved in DNA replication. We have already discussed about DNA helicase, single strand binding protein and primase. You can refer my video, the link is given in the description box. Today we are going to discuss about DNA topoisomerase. We have already discussed in detail that DNA has the double helical structure. Nitrogenous bases are hydrophobic and sugar phosphate backbone are hydrophilic. Hence DNA is twisted. This is due to the hydrophilic and hydrophobic interactions between DNA and fluid content of cell. We know that most of the content of cell is fluid or liquid. Hence nitrogenous bases are away from fluid, that is inside, because these are hydrophobic. Sugar phosphate backbone is in contact with fluid, that is outside, since they are hydrophilic. So as we have discussed in the previous video that this is the double helical structure of the DNA. These are antiparallel. These are the nitrogenous bases which are inside as they are hydrophobic, and the sugar phosphate backbone are outside, that is in contact with the fluid content of the cell, as these are hydrophilic. DNA replication depends on double helical nature of DNA. DNA molecule replicate or produces two identical copies. Each daughter cell that we get during the cell division receives a copy of DNA. And DNA replication is by semi-conservative mode. Among the two strands, one is the newly synthesized strand and the other is the template strand. During replication, two strands start separating at origin of replication. Further separation takes place by the helicase enzyme. As a result, replication fork is formed, which is Y-shaped structure. This is the origin of the replication. The two strands separate or melts at the origin of the replication. Then DNA helicase is loaded. Further separation of the strand takes place by the DNA helicase and Y-shaped structure is formed, which is known as the replication fork. As replication fork proceeds or DNA strands are further separated, torsion or tension, that is stress, develops ahead and behind of replication bubble. In order to prevent damage caused due to torsion or tension, supercoiling occurs. That is, change in the geometry of DNA arises, or we can say topological issues arise. So this is the replication bubble, and as it proceeds, tension or torsion develop. In order to protect it from the damage caused by tension or torsion, supercoiling of the DNA takes place. Ahead of the replication bubble, it overwounds, and it is known as the positive supercoiling. The direction of coiling is in the clockwise direction. If we see behind the replication bubble, here also supercoiling of the DNA is taking place. Here it is underwinding and the DNA is supercoiled in this direction, that is anti-clockwise, and it is known as negative supercoiling. We will discuss supercoiling of DNA in detail in separate video. Here we are going to discuss it briefly. So now what is supercoiling of DNA? It is twisting of the DNA double helix upon itself. So this is the DNA double helix and it is twisted upon itself here and also here. This is known as supercoiling of DNA. It arises in any helical or coil structure in which two ends are constrained. That is, when the two ends are severely restricted in scope. So when the two strands of the DNA is separated and their ends are constrained or it is restricted, then in order to protect it from the damage caused by tension or torsion, which arises, supercoiling of the DNA takes place. And the DNA can be overwound, which is known as positively supercoiled. It is right-handed. Or it may be underwound, that is negatively supercoiled. It is left-handed. So this is covalently closed circular DNA. It is in relaxed state. So when the torsion or the tension developed due to the separation of the strand, because this is constrained, then the DNA is twisted upon itself. It is known as supercoiling. Here it is positively supercoiled or overwound, and the direction of the supercoiling is right-handed or in the clockwise direction. Here it is negatively supercoiled or underwound, and direction of the supercoiling is left-handed, that is anti-clockwise direction. Now let us see some important terms of supercoiling. There are three important terms of supercoiling. First is the linking number, denoted as LK. It is the number of times one strand crosses over the other in space. Second is the twisting number, TW. It represents the total number of helical turns in DNA. Third is the writhing number, WR. It is the number of times double helix cross each other. It represents the supercoiling of helix. So if we see in this figure, this is the double helical structure of DNA. Number of times one strand crosses over the other is known as linking number. Here number of times one strand is crossing over the other is 1, 2, 3, 4, 5. So here this is the linking number. Twisting number represents the number of the helical turns in DNA. This is 1 turn, 2 turn, 3 turn, 4 turn. This is the twisting number. Writhing number is number of times double helix cross each other. Here double helixes crossing each other one time, and this is the second time. So writhing number is 2. We can represent linking number is equal to twisting number plus writhing number. In the relaxed DNA, there is no supercoiling, that is writhing number is 0. So in that case, linking number is equal to twisting number. There is the need to remove supercoiling or maintain the topology of DNA. Why? In order to express the supercoiled portion of the DNA. So the region of the DNA that is supercoiled, it cannot be expressed during replication or transcription. So in order to express the genes that are present in the supercoiled portion, it is important to maintain the topology of the DNA or to remove supercoiling. And the topology of the DNA is maintained, or the supercoiling of the DNA is removed by DNA Topoisomerase enzyme. This enzyme was discovered by James Wang in 1971. Now let us see about the topoisomerase enzyme. As the name suggests, these are isomerase enzyme. These helps in changing the DNA topology, or it maintains the DNA topology. It acts only on DNA strands. It relieves the excess twist, that is supercoiling, which accumulates during replication. It acts by transiently breaking and resealing the DNA backbone. Break that is caused by the DNA topoisomerase is not permanent, but it is temporary. Now the types of topoisomerase. There are two types of topoisomerases, type 1 topoisomerase and type 2 topoisomerase. So first is type 1 topoisomerase. It produces transient single strand break. It does not depend on ATP. It stimulates relaxation of supercoiled DNA. Now the mechanism of action. It cut the single strand DNA. Uncut DNA strand passes through the break. Then the religation takes place, that is cleaved strand is rejoined. Now let us understand this with the help of the figure. This is topoisomerase 1 enzyme and this is double stranded DNA. Topoisomerase 1 enzyme comes and binds with one of the strands of the DNA. It cleaves this strand of the DNA, and then the uncleaved strand passes through the gap of the cleaved strand. Then the religation or rejoining of the cleaved strand takes place. Now the DNA is relaxed. This is topoisomerase 1. There are two subclasses of type 1 topoisomerase, type 1A and type 1B. Type 1A, it forms a covalent intermediate with 5' carbon end of the DNA. It changes the linking number by 1. It relaxes negatively coiled DNA. Type 1B form covalent intermediate with 3' carbon end of the DNA. It changes the linking number by an integer, that is whole number. It relaxes both negative and positive supercoils. So this is the double strand of the DNA. Type 1a, it binds with the 5' carbon end of the DNA, and type 1b binds with the 3' carbon end of the DNA. Second is type 2 topoisomerase. It cut both strands of the DNA at once. It is ATP dependent and it changes the linking number by 2. Mechanism of action: it cleaves both the strands of the DNA. Intact strand passes through the gap, and then the religation or the rejoining of the cleaved strands takes place. Now let us understand the mechanism of action of topoisomerase 2 enzyme with the help of this figure. This is the region where two double helices cross each other. At this site, the topoisomerase 2 enzyme is activated. It requires energy for its activity, which is obtained by the hydrolysis of ATP. So, it binds at this site. Then one of the double-stranded DNA is cleaved. It cleaves both its strand, and the other DNA helix, which is intact, passes through the gap. Then this cleaved DNA is rejoined or religated. In this way, supercoil is removed or DNA is relaxed. Let us understand the action of topoisomerase 2 enzyme this way. This is the supercoiled DNA. Topoisomerase 2 cuts both the strands of the DNA, and then the intact DNA passes through this cleaved region or the gap. Hence, two supercoils are removed at a time, and then this cleaved region is sealed. This is the relaxed DNA. Hence, topoisomerase 2 changes the linking number by 2. There are two subclasses of type 2 topoisomerase. These share similar structure and mechanisms. First is type 2a. It is found in viruses and all cellular organisms. It is of three types. Topo 2 found in eukaryotes, Topo 4 found in bacteria, and gyrase, which is found in bacteria, example E. coli, and in some eukaryotes. It introduces negative supercoiling, decreases the linking number by 2. Second is the type 2b. It includes Topo 6. It is found in archaea and some plants. Type 2 topoisomerase or topoisomerase 2 has two subclasses, type 2a and type 2b. Both are similar in structure and function. These are heterodimeric. These have two monomers. One monomer binds to one of the DNA strand and the other binds to the other DNA strand. And then these cleaves both the strands at a time in case of type 2a and also in type 2b. Now the topoisomerase inhibitors, chemical components that suppress the action of topoisomerase are known as the topoisomerase inhibitors. Their inhibition principle or topoisomerase inhibitors are used in the development of drugs in order to protect us from bacterial infection. It includes antibiotics as novobiocin and conmermycin, and these suppress the activity of topoisomerase enzyme of bacteria. So this is all for today's video. In the next video, we will discuss about DNA polymerase enzyme. So stay tuned. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.